Hello everyone, Sangeeta here. Uh, let me discuss on fatty acid synthase complex. This comes under the revision on lipid metabolism. So from this topic, three questions have been come. The first one is fatty acid synthase complex, which carries biomark. Then mention the reaction catalyzed by ATP citrate lyase, which is for 1 mole, and mention the rate limiting enzyme in fatty acid synthesis. Question number one, fatty acid synthase complex. So what is it exactly? So this is the multi-enzyme complex enzyme system which mainly help for the de novo biosynthesis of fatty acids. So in humans, fatty acid synthesis occurs mainly in liver, lactating mammary glands, and in some extent in adipose tissue, kidney and brain. So de novo biosynthesis means new synthesis of amphibolic compounds. That is the meaning of de novo synthesis. So this fatty acid synthesis which occurs from the precursor acetyl coenzyme. And we know that the end product of fatty acid oxidation also acetyl coenzyme. But it is not exactly the reversal of beta oxidation, this de novo biosynthesis. So the fatty acid synthesis occurs in three stages. First one, transport of acetyl coenzyme from mitochondria to cytoplasm. And second, carboxylation of acetyl coenzyme to malonyl coenzyme. And the third, reactions of fatty acid synthase complex. So our question is, what is this fatty acid synthase complex? So in detail, this fatty acid synthase complex is a dimer of two identical polypeptide monomers 1 and 2. And the molecular weight of this fatty acid synthase complex is 2,40,000. And each monomer, which consists of 7 different enzyme activities and an acyl carrier protein with phosphopantothene SH group, pantothene sulfhydryl group. And it consists of 3 units. One is condensing unit, second one is reduction unit, and third one is releasing unit, or it can say as domains, first domain, second domain, and third domain. So this is the overall figure of fatty acid synthase multi-enzyme complex. If the question comes, you are supposed to draw this figure for multi-enzyme complex. So you can see that it has same monomers in both direction. It has two division. One is a subunit division like this, and it has another division that is a functional division. Okay, so you can see that it consists of condensing unit, releasing unit, and reduction unit. Reduction unit and releasing unit. Okay, so the condensing unit consists of the enzymes ketoacyl synthase, acetyl transacylase, malonyl transacylase, and this ketoacyl synthase consists of a sulfhydryl group in its structure. And reducing unit, which consists of enzymes dehydratase, enoyl reductase, ketoacyl reductase, and acyl carrier protein, which is attached to a sulfhydryl group through 4 prime phosphopantothene group, and the releasing enzyme is thioesterase. So this subdivision is known as subunit subdivision, but for the proper functioning of fatty acid synthase complex, it always should have two monomeric unit, and first half of condensing enzyme and the second portion of the reduction unit together only can carry forward the fatty acid synthesis. So this fatty acid synthase complex has two active sites. Active sulfhydryl group in its structure. One is attached to the acyl carrier protein, another is attached to the ketoacyl synthase. And we can see that it has a head to tail arrangement of the monomer unit. And actual functioning unit consists of one half of one monomer interacting with the complementary half of the other. Now the significance of this fatty acid synthase complex is that it has greater efficiency coded by a single gene and good coordination for the synthesis of all enzyme. So each monomer consists of enzyme activities of fatty acid synthesis, So the intermediates of the reaction can easily interact with the active site of the enzyme. So these are the significance of fatty acid synthase complex. Now, the individual role of these three units, condensing unit, reduction unit and releasing unit. So already got familiarized with enzymes. Condensing units consist of three enzymes. Condensing enzyme, which is also known as beta keto acyl synthase, acetyl transacylase or acetyl transferase, and malonyl transacylase or malonyl transferase. The role of this unit is that which initial substrate bindings. This is the initial substrate binding site for the fatty acid biosynthesis. Second is a reduction unit, which consists of the enzyme dehydratase, enoyl reductase, keto, beta keto acyl reductase, and acyl carrier protein. The function of this unit is that the ACP, which is a polypeptide chain having a phosphopantothene group, to which the acyl groups are attached in thioester linkage. That is a function of reduction unit. And releasing unit consists of the enzyme thioesterase or deacylase, which mainly help for the release of newly synthesized palmitic acid, which is a predominant fatty acid synthesized by the human body. So, you should draw the fatty acid synthase complex properly and briefly explain the structure and write the significance of these fatty acid synthase complex, why such an arrangement, how it will help for the efficient synthesis of fatty acid. Then individually you have to write the three unit and its functions. Second question is, mention the reaction catalyzed by the ATP citrate lyase. So we have to think where this ATP citrate lyase enzyme have come. So the role is that in second phase, that is first phase transport of acetyl coenzyme from mitochondria to cytoplasm. So the acetyl CoA, which is a precursor for fatty acid, is available in the mitochondria. So it has to reach the cytoplasm, but the mitochondrial membrane, which is impermeable for the transport of this acetyl CoA. So it has to end in the form of another thing, that is citrate. So for that, the acetyl coenzyme combines with oxaloacetate by citrate synthase enzyme and forms citrate. This citrate reaches to the cytoplasm where resynthesis of acetyl coenzyme occurs, and this acetyl coenzyme will be utilized for the fatty acid synthesis, isn't it? So inside the cytoplasm, the enzyme ATP citrate lyase, which catalyzing citrate to acetyl coenzyme and oxaloacetate. So this reaction you are supposed to write for the 1 mark question. So while writing, you can write citrate plus ATP plus coenzyme A gives rise acetyl CoA plus oxaloacetate, and the enzyme is ATP citrate lyase. That is the way you are supposed to write. Third question, mention the rate limiting enzyme in fatty acid synthesis. Your answer should be acetyl coenzyme A carboxylase enzyme, which is the key enzyme or rate limiting enzyme for the fatty acid biosynthesis. Thank you.